not exactly amino acid, they are amino acids. So certainly proline and hydroxyproline will not be detected by a common test, which is normally for amino acid. So for that one we are having a specific test that is called as Isatin test. So let us understand what is this Isatin test. So this is a biochemical test by which we are going to detect two specific amino acids, that is proline and hydroxyproline. These are the not exactly amino acid, here it's written amino acid, commonly we are calling it amino acid, but exactly it is a amino acid. So this test is a specific test for a specific amino acid that produces a colored addition product, which we are going to call adduct. Adduct means addition and product with the Isatin reagent. Here we are having, if you see the structure of proline and hydroxyproline, we are having proline. This proline, after the hydroxylase, it is going to be converted here. OH group will be added here and it becomes hydroxyproline. So why it is not the amino acid? This is the amino acid. If you see in amino acid, we are having normally COH group. This is the central carbon we are having, and this central carbon normally we are having NH2 group and R group. In case of proline and hydroxyproline, this amino group and R group is going to form a ring structures. So that's why it is not the exactly amino acid, it is called as amino acid. So the detection of the amino acid proline for a long time had been unsatisfactory, as there are methods like the ninhydrin test can only detect proline for isolation from this amino acid. And if you see the ninhydrin test, there we are going to have Remans purple is going to form for the amino acids. Proline and hydroxyproline normally gives you a different types of yellow color pigment colored product in case of ninhydrin test also. So there also proline gives you a different type of tests. Now later in 1971, Fouad and Boctor came up with a reaction between the proline and Isatin, which can be used qualitatively as well as quantitatively for the determination of proline and hydroxyproline, and this is called as Isatin test. The distinction between the proline and hydroxyproline cannot be made by this test. So we can, proline and hydroxyproline both going to give you the same type of process, same type of reaction. So you cannot differentiate proline with the hydroxyproline, rather you can differentiate other amino acid from the proline or hydroxyproline. So the objective of this test is, there are two specific objectives. One is you can detect the presence of proline and hydroxyproline from in the protein sample or in any sample, and second, you can differentiate proline and hydroxyproline from other amino acids. What exactly you require for this particular test is, so in this case, because we are going to use Isatin, so we require a specific reagent that is called as Isatin reagent. What is this reagent? It is a 0.02% solution of Isatin prepared by dissolving in Isatin crystal in 0.5 molar sodium citrate buffer at the pH ranging from 3 to 11. And then we require a test sample which we are going to detect it, and then we require a concentrated hydrochloric acid. This is the structure of Isatin. If you see this structure, this is somewhere how it looks like ninhydrin also. What are the material we require? We require a Watsman filter paper, hot air oven, and pipette we require. If you see the principle of this test is, the basic summary of the principle is, Isatin reacts with proline and hydroxyproline and give you a specific color product that we are going to see it. So in details, if you see it, Isatin reagent used in this test work as a visualizing agent which provides a different color with different amino acids in the chromatographic techniques. When we are performing chromatography, that time every amino acid gives you a specific kind of test. The Isatin reaction between the proline and hydroxyproline gives you a blue color product, and the reaction between amino acid like proline and hydroxyproline and Isatin, that is 1H indole 2,3 dione, yields a blue color products. So this blue color product presence is means it is a kind of confirmation that there is a proline presence or amino acid is present there. So the reaction is a simple addition reaction where the combination of Isatin and proline produces a colored product indicating the presence of the amino acid or presence of proline and hydroxyproline. How we are going to perform? This is again a simple test where a drop of the sample, that is amino acid solutions, is added to a Watsman filter paper and dried by placing it in a hot air oven. So now we are going to drop Isatin reagent added to the dried spot, and the process of drying into the higher air oven is, hot air oven is going to be repeated. Means first you put your test sample, dry it, and then put the isatin reagent, again dry it, and the paper then you can going to observe for the formation of blue color spot. If blue color spot forms, it means it is a positive reaction, amino acid is present. If blue color spot does not form, it means it is a negative test. So positive reaction means positive result is the Isatin test is represented by the formation of a blue color spot on the filter paper, which indicates the presence of amino acids, means proline and hydroxyproline. And negative result means when you don't have no pink color spot on the filter paper, means no blue color rather than any other color formation will be occurring, because you have already seen that this isatin gives you different types of color with the different amino acids. So blue color with the proline and hydroxyproline, any other color with other amino acids. So if blue color does not form, it means there is a no amino acid, it is any other amino acids will be there. So this is the process steps and result. We are going to have Watson filter paper. From this one we have taken a sample. On this filter paper we have kept it. We are going to drying it into the hot air oven. Then we are going to put one drop or few drops of isatin reagent, dry again into the hot air oven. Then you are going to see which type of spot visualization will be there. If blue color spot will forms, it means hydroxyproline and proline are present. If pink color spot or any other color formation is there, means amino acids are absent or any other amino acids can be present. So again, if you see the usage of this test, there are two specific use of this test is. The test is used for the detection of the proline and hydroxyproline in the test sample, and Isatin test is one of the standard tests for the detection of polynuclear hydrocarbons and phenol in the polluted airs. The method is simple, fast, and suitable for the estimation of proline content in the larger sample. Also, this test can be used as a qualitative as well as quantitative test for the proline and hydroxyproline or any other amino acids. And there are certain limitations for this test is the color observed in the test should be observed carefully, and the test should be repeated if the mixed color is going to be observed during any of the tests. So this is the limitations. This is all about for Isatin test for proline and hydroxyproline. Slowly we are going to cover all the qualitative and quantitative tests for all the amino acid and other biomolecules. So this is all about for today. Hope you have understand it. If you have any query, any comments, you can write in the comment box. Another news for you, I want to share with you is that thanks button has been activated on my channel. So if you really like the video, if you like the content, you can, if you want to support us, you can click on the thanks button and you can support us. So, and other than that one, you can follow me on any social networking sites, social networking platform. You can write your questions, you can write your comments. Thank you very much. Have a nice day.